Hey, hi everyone. Welcome back to my channel, Agriculture UPSC Optional Simplified. So in the last video, we have discussed the subjects and the syllabus keywords of paper 1. In this video, I'm going to discuss the paper 2 syllabus and its subjects. So without wasting any time, let us go through the subjects. So here are the subjects that you can see. So the first thing here is plant genetics and plant breeding and its seed technology, plant physiology, horticulture, pathology, entomology, and food technology. Don't just panic if you are from non-science background or non-agriculture background. I'll teach you everything well, so no need to worry. Don't just panic just by looking these words. And so let us see the syllabus of each and every topic here. The before that, I just want to say one thing. So comparing paper 1 to paper 2, and it is very common with other optionals too, that paper 2 is bit difficult, and it's not always paper 2 among all the optionals, but if we compare from paper 1 to 2 of any other optional, one paper is going to be really tough, and in this agriculture optional, paper 2 is going to be tough compared to paper 1, and there are many reasons behind that. The first reason here is the first thing, the paper 2 is totally technical in nature. So it's totally technical. There is one answer fixed for this question that is asked in the examination, and we don't have any such kind of scope writing in our own way. We have to know the concepts clearly, otherwise we can't write the answers, and this is one such hard thing that we need to face. And few other things, in paper 1 we can align some current affairs, some our knowledge into some subjects like farm economics or farm extension, but we only have such kind of chance related to this food technology, and all other subjects have the technical answer that is fixed to it for each and every keyword in the syllabus. But on the other side, it is easier than other subjects because answer is already fixed to what keyword, and the same keyword is going to be asked in the examination. So it is easier for us to guess, remember, frame the answers, and save a lot of time. So this is the advantage too. So let us see the syllabus now. Genetics. The first subject here is genetics. The first thing here is cell structure. First of all, what is genetics? Same like humans, it's better to compare with humans so that it will be clear for us. So we are a human having the characteristics from both our mother and father, and we use the basic word here called genes. Sorry, genes. So this genes is what we draw from our parents. So that is the reason why we have both mother and father characteristics. How this genes is transferred from them? This genes is transferred from DNA. That is why we do DNA test to test whether the child is related to this particular parents or not. So this, from this DNA we are getting the genes from our parents. In the same way, this genetics deals with the plant's genes that comes from generation to generation. If we observe the wild banana, whatever the banana that we are eating nowadays is not the original one. So after many changes, after many breedings, it changed to the present day banana, which is having a lot of flesh in it with a very tiny seeds which we can chew, and that is not visible sometimes. So it is easier for us to eat easily. But if we see the wild banana which is once there long days back, the flesh is very less and the seeds are more. It is difficult for us to extract this flesh. So in such way, there are many crops like maize. If we take maize, is also having a very less seeds on these pods. But nowadays we can see the maize which is bigger in size, having a lot of seeds, means a lot of yield we can get. So the characteristics from one parent, one plant, and the characteristics that the good characteristics from many plants combined together and produced the plant that is desired for us. Only if we know the characteristics and genes of these plants, we can breed the way we want of the particular crop. So that is the reason why learning this genetics is very important. So first thing that we see here is cell structure. So what is cell? Even the humans, we are a cell first. We are not a visible structure first. We are a cell. So after this cell, we formed like zygote, embryo, and many other things, and we form a full body now. So in the same way, plant do also have a cell. So the cell constitutes different parts in it, and everything has a particular function for that, and all together having these functions make a cell grow, and making it grow as a plant or leaf or something related to the plant. And next, what is the function of this cell? And next, what is the cycle of this cell? All these things are somewhat conceptual in nature. Don't worry, I'll make every concept clear to you just by making you to go into a different world of this plant. So almost everything that we study in genetics is not visible. We have to go into deep. For example, cell is what something deep in this leaf. Leaf is visible to us, but in deep down there is a small thing. Combination of cells constitute this whole leaf, and all these cells doing its own functions to make this leaf function to grow, helping growing the plant. So we should know the structure and its function and in cycle. The next thing is synthesis, structure and function of genetic material. So in cell there is this genetic material which is RNA, same like DNA. So this we should understand how the synthesis takes place from this cell cycle, and what is the structure. We can see out of this DNAs or RNAs, there is this segments called chromosomes. Altogether forms these long strides. So we have to study the chromosomes. That is nothing but the genetic material. And then next, loss of heredity. And this is a separate section, different loss of heredities. And next, chromosome structure. So as I told you, there are chromosomes in such way, and we need to study the structure of that. And aberrations means nothing but the changes, and linkage, crossover, and their significance in recombination breeding. How all these things helps in breeding. For example, I'll tell you in a very simple way. This is a chromosome of one plant. This is a chromosome of one plant, and it is having genetic material in it. So characteristic of one plant. And let us take the chromosome of different plant. It is having a different characters. Now we need to breed one new thing based on the characteristics that we deserve. So we have to take small segment from this thing, and we have to take a small fragment from this thing, and we have to combine these both things. Together we can produce one new thing which is desired for us. So all these things and how it is helping in recombination breeding process is what we need to study. Next, polyploidy, euploidy and aneuploidy. These three terms are very famous and the questions are very direct from these things. Same like cell structure, the question is very direct. What is cell? What is the function of the cell? What is cell cycle? Explain the different types of cell cycles. And the question is very direct. What is polyploidy? What are chromosomal aberrations? What are the types of chromosomal aberrations? So they will ask the very direct question, taking the term from this syllabus. See, this is a term. They will directly take this term and ask in the examination the question, what is chromosomal aberrations and what are the types of chromosomal aberrations? Explain with diagrams. This is what they will ask. And next, mutations. Mutations means nothing but the changes and the role in crop improvement. And next, heritability. This is one term. Sterility and incompatibility. So all these things has a special purpose for breeding and classification and their application in crop improvement. What is the term meaning crop improvement mean? Crop improvement means nothing but getting the desired crop for us. Same like I explained you here by taking the example of banana. So that is nothing but the crop improvement. So all these things will help us. And next thing is, I'll make everything clear with a lot of examples while I teach you all these things. Don't need to worry about that. But as a glance, I am explaining you. Cytoplasmic inheritance. So this is a common term and famous term that we can see direct question from this. What is cytoplasmic inheritance? Sex linked, sex influenced and sex limited characters. And these three things, out of these three things, sometimes we can see the questions too. So this is all about the genetics. And after understanding this genetics, we can understand this plant breeding very well. So what is meant by breeding? Producing something new. So here what we are breeding? We are breeding the plant, desired plant, by taking two different plants, combining these two plants and producing a new thing, new plant that is desired for human consumption, which is having a lot of benefits like higher yield and many other things. Let us go through one by one now. History of plant breeding. How this plant breeding has started, who are involved in that, and what are the procedure that they have followed, and modes of reproduction. So normally if we take flower, so we can find this anthers, which is male male related, and we can see a lot of structures, female related, male related, and we can see the structures like anthers, pistil and many things. Everything has a purpose, and there were different modes of reproduction will be there, like uh, dioecious plants, monoecious plants, and how other organism, wind is helping with the pollination, and how the animals are helping, how butterflies are helping. There were different things that we'll be discussing detail in these modes of reproduction. Selfing and crossing techniques. So selfing is nothing but within this plant how the crop is reproducing. For example, like if we take this anther, the male part, the same flower, the pollen from the same flower is reproducing this female part. So together they forms a full fruit. And crossing means from other plants. So there were different types of sections in this selfing and crossing techniques that I'll discuss clearly. And origin, evolution, domestication of croplands. So where the particular crop, whatever the crops that we eat now, like rice, its original habitat is at one place at first. Maize, maize or some other like sorghum, it is from Africa or somewhere, and maize is from Mexico. So in the same ways, rice is from Asia. So their original habitats are from different parts of this world, so their origin is different. We brought them and introduced it at the new place, and their evolution, I mean, we just made those type of plants suitable for these climatic conditions in other countries too, and we domesticated them based on the requirements that is suitable for that plant and rebreed it according to that. And its center of origin, law of homologous series, and crop genetic resources conservation and utilization. So how we should conserve the new plants and how we should utilize them properly is what we study here. And next thing here is application of principles of plant breeding. So while we breeding the plant, what are the principles that we need to follow? And improvement of crop plants is what we study. And molecular markers. So while breeding, there are lot many process that we need to go through, and these molecular markers are one such type of thing, and their application in breeding process. Pure line selection. We have different types of breeding where we can see here pure line selection or pedigree, mass, recurrent, combining. So all these things are types of breeding, and these things are different from each other, so which is suitable for different crops. We should study all these things and having a similarities too, so it is easier to understand. Heterosis, this is also a type of thing of breeding. And somatic hybridization, we all be knowing familiar with this term called hybridization, and everything that we eat nowadays is a hybrid varieties, so which is a combination of different parents. And what is somatic hybridization is what we study here. And breeding for disease and pest resistance, so there were certain kind of plants like bt cotton or bt brinjal. So in such way there were certain kind of varieties that has been produced, especially some varieties are produced to this resistant to these diseases, so automatically no disease will come. And pest resistance also, for example, few plants, for example rice. If we take rice, the stem borer is a common pest here, like it will directly suck the liquid content in the plant outside. But now what they have breeded is, so they have the spikes over the stem, so it is difficult for the stem borer, it is difficult for the stem borer, sorry, just a second, to suck the liquid content from that stem. So these are the types of varieties that they have provided. And next thing is role of interspecific intergenic hybridization. This is also one type of hybridization that we can see. And role of genetic engineering, biotechnology, how they are important and how they are contributing in crop improvement, that we can see. Genetically modified crop plants, and these things are commonly asked in examination nowadays in the science and tech part and this plant breeding too. So we have to study what are the genetically modified crops until now and how they are providing produced until now is what that we see.